research professor at UC Santa Cruz, and his interests are the origin of DNA, RNA, protocells. He's done a lot of work over the years on membranes in particular, and polymerization from the monomers to the polymers. And he's been developing an idea about that some hydrothermal pools on land could be conducive to the assembly of life. He also wrote a nice book in 2019 called Assembling Life, Oxford University Press. So if you want to learn more, you could read his book, but he's going to tell us about his hydrothermal fields today. So
thank you.
Please go ahead.
Thanks kindly. It's a real treat to be here. This is the first time I've done this in 2 years. The last time was in Australia, and I was at the University of New South Wales just before pandemic came through. And of course, that really put a big gap in all of our lives. But here we are again. Let's see if I can get rid of that continued thing. There we go. Hydrothermal fields are conducive to assembly of nucleic acids and protocells. Well, that's a pretty big claim, and it is a little bit on the controversial side because there is a competing idea, partly developed here at this campus, that hydrothermal vents are a conducive place for life to begin. So I like to think of these two as alternative hypotheses. And that's a kind of a gentle way to say that, yes, you've got just a different idea than I do, but instead of arguing about the ideas, let's go out and test them. If you have alternative hypotheses explaining the same observation, that's a little bit better. It's better than saying that I'm going to go out and prove that I'm right, that my hypothesis is right. Because most ideas like this are wrong when you actually try to map them onto the real world. So the idea is to get rid of the ideas that are wrong by weighing the evidence. And in a sense, it's like a jury. So I'm one of the prosecuting or defending lawyers, and you are the jury today. You're going to listen to what I say. You're going to weigh the evidence with me, and I will tell you why I think that hydrothermal fields are conducive to the assembly of nucleic acids and protocells. Now, in principle, this little guy should change the slide. I might not be able to make that work. I'm sorry?
You may have to click on your slides because you clicked on the view box.
Okay. Well, let's see. I'm now clicking on my laptop forward, and all I get is a little melodious bump.
I think I know what you mean. May I try?
Thank you, Sarah.
You're welcome.
There. Okay. What did you do so I can do it?
I followed their notes. If you click on the your presentation,
Yeah.
after the Zoom box, then it'll return to your presentation.
So
We think we got it. You should be okay going forward.
I'll play around with it. Well, I wanted to show you a hydrothermal field. You've seen lots of hydrothermal events if you're here and have been listening to all the wonderful work being done by John Ross and Deborah Kelly and Jody Deming. This is a good idea. I'm not dismissing it, but I'm saying it is a hypothesis at this point and needs testing. So the alternative hypothesis is what you're seeing here. This is a photo I took in Kamchatka, Russia in 2004. And that was a field work that gave me the original idea on how life could begin in a hydrothermal pool, because I looked at pools like this. They were hot, they were bubbling. You can see some bubbles there. They obviously were on the way to drying out as long as it didn't rain again. So these things were going through cycles of wetting and drying. of hydration and dehydration. And I'm going to claim that that physical cycle of wetting and drying can cause polymerization to happen and lead to polymers such as nucleic acids. Yeah, that works. So I want to tell you some advice that my major professor gave me when I was coming up for my PhD. And he said, when you're giving a talk, and this is for the young people in the audience, when you're giving a talk, you start by telling them what you're going to say. And then you tell them, and then you tell them what you told them. So I'm going to follow that advice today, and I hope that you can take it too. I'm going to tell them what I'm going to tell them. So that's easy enough. Life can begin in freshwater hot springs. I'm going to make that argument as a hypothesis. There's chemical energy in wet-dry cycles. Now, you might think that if you dry something out, that's all that happens. But as a matter of fact, what really happens is you change the entropy of the system and the enthalpy of the system in ways that are favorable for condensation reactions to occur. What that means is that thermodynamically, I should be able to cause polymerization to occur just by wet-dry cycles. And if I really do cycle it again and again and again, I can drive the complexity of the system thermodynamically uphill away from equilibrium. And that is one of the main lessons that you're going to learn as you listen to this talk. Chemical energy in wet-dry cycles. I'm going to use nucleotide monomers. These are the 4 monomers of RNA and of DNA. For RNA, it'd be adenosine monophosphate, AMP, the uridine monophosphate, UMP, it'd be guanosine monophosphate, and cytosine monophosphate. Those are the 4. And DNA is the same, except they're deoxy mononucleotides. Instead of uracil, it uses thymine. So nobody knows exactly why that happened, but evolutionarily is a fact of life. So those are the 4 monomers. And I'm going to see whether I can get them to polymerize using the simplest energy available on the early Earth. When you think about it, just wet something, dry it out, wet it, dry it out, and you're going to see whether it works or not. Finally, encapsulated polymers are something I call protocells. So if I have a membrane-forming lipid present in this mixture, it's going to form vesicles. And if I have made polymers, those polymers are going to become encapsulated in the lipid membrane. And we refer to those as protocells. They're not alive, but they are a very important step toward life. The reason is that every protocell is different from all the other protocells. They're made by random processes of self-assembly, so they're all different. What that means is that each protocell is a microscopic experiment trying to hang around, trying to be selected by the stresses of its environment. So sturdier protocells, more robust protocells will have a property that allows them to be selected. And that, I think, is the first step of Darwinian selection, is selection of protocells for stability, for having some sort of porosity in the membrane, and finally, for containing some catalysts that are important for a primitive metabolism and so forth. So you're going to see that I can do some of those steps in the laboratory and out in the field. We're taking our experiments into hot springs out in the real world. We're seeing whether this works under natural conditions. And this last thing is just that populations of protocells can evolve. So there you go. That's what I'm going to tell you. Now here's some examples of hydrothermal conditions. The ones you've heard about are black smokers, discovered by Jack Corliss in 1977. John Burroughs and his grad student Sarah Hoffman, right here at University of Washington, suggested just a few years later that not only does microbial life live in these conditions, but life may have begun there. That idea caught hold, and it has been around now for 30 years or more, as people have just said to themselves, the oceans are 99% of all the water on Earth. Of course, life must have begun in the ocean. So that's what we are countering in our work. We're saying that maybe, maybe not, but let's think of another place as well. How about freshwater? 1% of freshwater that is falling on any landmass arising above a global ocean. So that is the contrast between what has been proposed in the past, the paradigm that life began in the ocean, and what we're proposing now, that maybe there is another place as well we should consider. So black smokers, and then Deborah Kelly led a team that discovered alkaline vents. The black smokers depend on magma, hot magma, for their energy. The alkaline vents depend on a chemical reaction called serpentinization. Seawater reacting with olivine minerals produces another mineral called serpentine, but it also produces very alkaline liquid, which comes up and precipitates as these mineral columns that you see there in the middle. And most importantly, for that story, there's a lot of hydrogen, gaseous hydrogen, dissolved in that water. And a big theory has been set up by Mike Russell and Bill Martin, Nick Lane, that somehow that hydrogen can provide reducing power so that carbon dioxide can start into the organic compounds required for the formation of life. It's a valid hypothesis. It is being tested. I doubt that it'll ever be tested down at the actual events. That's going to be a very expensive experiment, but at least people are trying it out in the laboratory. Clear to the right is Bumpus Hell. Bumpus Hell is named after Mr. Bumpus, who was guiding some reporters to this site in the mid-1800s. And he accidentally stepped through a mineral crust and buried one of his legs up to the thigh in boiling mud. He walked out, got on his horse, and rode home. They were tough back in those days, but he lost his leg. So his name now is associated with this. It's in Northern California on Mount Lassen. It's the remains of an eruption of Mount Lassen back in the early 1900s. This is really a nice field site. A lot of people are using it for various reasons, and we've done experiments there. So keep in mind that the alkaline vents are in salty water, and hydrothermal fields are being fed by distilled water in the form of precipitation, distilled from the salty ocean and falling as rain or snow in the case of Bumpus Hell. So those are the two contrasting kinds of water that we're talking about. Well, I wanted to see what the prebiotic Earth really looked like. And what you have to do is to say, well, there were probably volcanoes on the prebiotic Earth. That's what the geologists tell us. We can see fossilized volcanoes on Mars. So Mars had volcanoes. Probably the Earth did as well. So I'm just taking it for granted that there were volcanic landmasses, but there was no continents as we know them now. The plate tectonics had not started up yet. Okay, so to see my first introduction to the prebiotic Earth analog site, I flew from San Francisco to Anchorage, Alaska. And you may remember that's where Sarah Palin said she could see Russia from. I'm not sure about that. And then next morning, we would get on Magadan Airlines to Petropavlos, this little bay on the southern part of Kamchatka. Petropavlos sounds like a very Russian name, and it is Russian, but it's Russian for St. Peter and St. Paul. Vitus Bering, who discovered the bay, gave it the name after two of his ships, St. Peter and St. Paul, Petropavlos. This is what it looks like from a satellite view. This is an artistic rendition of a satellite view. And what you see there is a line of volcanoes. This is the most volcanic region on Earth. So that's why it's a good place for people like me to visit. You see that we land at Petropavlos, and then just south of Petropavlos, within maybe half a day's ride is Mount Monoski, and that is an active volcano. This, by the way, is what Petropavlos looks like from the airplane as you're coming into land, and you can see the Koryatsky volcano just looming up behind it. So we got on a Russian troop carrier and took a dirt and gravel road down to Mount Monoski. And there it is. That's the mountain from a Google map of it. You see that little winding white thing? That's a gravel road that gets people down to Mount Monoski. It's a little bit of a tourist attraction now. But it is an active volcano. Occasionally it will erupt every few years. Not a great big eruption, but certainly you would not want to be in the cone when it erupts. So this is what our second expedition looked like. Believe it or not, that's a group of scientists. They don't look like scientists, but that's what we look out in the wild. I'll introduce two of them. In the red jacket is Jan, and the black jacket is Jake, and they were postdocs from the Carnegie Institution of Washington. In the light blue jacket is Jamie Elsila, and she is now a research scientist at Goddard Space Flight Center. She and Megan at the time, that's the young woman in the scarf, were grad students in chemistry at Stanford University. In the white jacket is Vladimir Kompenchenko. And if you ever want to go to Kamchatka, you better get somebody like Vladimir to help, because it's a different culture. There's a lot of fun. Is that me? No? Okay. A lot of strange things you have to do with money and exchange rates, and he knows how to do that, and we don't. So he took care of us during the trip. Clear to the right is Igor. There really are Russians named Igor, and he was our local guide on Mount Monoski, showing us where the hot spots were. And clear at the other end is my buddy, my friend, Tony Hoffman, from down the road where I live in Bonnie Doon, California. And he was our unofficial photographer. So the photographs you see are his. Here we are, that's Jan and Jake climbing this ice field on the edge of the crater. And as we got over that edge and looking down into it, this is my vision of the prebiotic Earth. Sterile, nothing is alive there in the usual way of plants and so forth. There's certainly microorganisms, but not much else. There is a lot of heat there. That's a glacier melting into the heat and putting up great clouds of steam. And we could hear the roaring from a distance as we came over the top. So we climbed down through all those rocks that you see, and here I am down in the crater, which has since been buried, by the way, by another eruption. And I'm taking a sample that I had taken on my first trip. I took that sample back to Santa Cruz to analyze for any organic material, because the Russians had published papers saying there were amino acids in this pit. And we didn't find anything like that, even at the parts per billion analysis rate. So I knew that there probably was not going to be organics in this boiling mud pit, as the Russians had claimed. So I took my own organics along on this second trip. We found a little boiling puddle. It's about 10 liters of boiling water. You can see the boiling action in the middle. And all around is the turbid clay of this boiling water. And you can see down at the bottom, we took a sample from the clay to begin our research. We took that sample home. And thank goodness we did, because that turned out to be quite important. Here I am with my experiment. Now, the experiment was to pour a prebiotic soup into this boiling water and just to see what really does happen to organic compounds if they could be introduced into a pit like this. Low pH, pH 3, because of the sulfur dioxide in all the water. There's, I put in some phosphate, some glycerol. That white that you see is a amphiphilic molecule, a 14 carbon fatty acid. We know that it can assemble into membranes, and that's why we put it in. We thought that maybe it might make a phospholipid if it reacted with glycerol and phosphate. Then we put in all 4 nucleobases, or RNA. We put in 4 amino acids. These are the 4 that are prominent in the Murchison meteorite and in Stanley Miller's famous experiment. So that's what went in, and we put in gram quantities. So we put in so much that any traces we just didn't have to worry about. We're really most interested in what happened there. Now this turned into a paper. It was published. I don't have time to go into all the details. I just have one result from this I'm going to show you. But if you're interested in what happens, what really happened, I can summarize in one sentence. Everything we put into the pool except one compound, everything disappeared from solution. We could not find it when we took it home and analyzed it. So it bound up to the clay. So that was a real lesson learned. Organic compounds bind to the clay very strongly. And we could only release it by bringing it up into an alkaline pH range. And we could see it then all come back off the clay. The one exception was this stuff. See that white material around the edge of the pool? That appeared within minutes. And if you think about it, a fatty acid is a soap. I added a soap to this pool. You would expect the soap to form a kind of a froth. But what I didn't expect is that froth would gather around the edge of the pool and that pool can dry out. If that happened, this is the idea. If that happened, lots of reactions might happen around the edge of that pool in the dry, concentrated reactants on the mineral surface. So that is what I'm really the start of what I'm talking about today. So just keep that in mind, that wet-dry cycles can do things that cannot occur in solution chemistry. So here are the favorable properties. This is what I know now. Organic compounds accumulate in these ponds in contrast to ocean. If this stuff down the ocean becomes diluted down to micromolar ranges. Acidic pH range, that's conducive for the kind of polymerization that we're looking for. Very low concentrations of ions. Keep in mind that these pools are being flushed every year by precipitation. So the only ions that get in are the ones that dissolve off of the minerals that the pool is in. Wet-dry cycles concentrate potential reactants, as I said before. Polymerization is promoted. You're going to see that a little later. Elevated temperature gives activation energy. That pool is about 80 to 90 degrees C. So our simulation is also in that temperature range. Finally, if there are membranous amphiphiles, they're going to assemble into vesicles and capture any polymers as protocells. So that should all happen. And the experimental stuff I'm going to show you is a test of this hypothesis. This is a testable hypothesis. This is kind of an image we made up just to illustrate this. Here's our volcanic landmass with delivery of extraterrestrial organics from infall. They accumulate in the pool and go through the wet-dry cycles. Here's a video. Now, this is a little cartoon we made up, a presentation of just what we think this would look like if we were back 4 billion years in time. So there would be steaming hot springs. There would be geyser activity. The splashes would fall on the camera lens. You can see the water running downhill toward the ocean. If life began in a hydrothermal pool, it would be delivered to the ocean downhill from here. That brown stuff is organic material floating on the surface. It flows through multiple pools. These pools can be different from one another in terms of pH and organic content. Then if you focus on the edge of a rock, as we see in this video, you can see that the water is going to be going up and down, lapping up and down as that geyser erupts. And that is a wet-dry cycle that happens on the edge of a rock like this. So it doesn't have to be periodicity of rainstorms. It can just be the wavelets wetting and drying on a hot surface. Okay, now let's talk about how we might get life to begin in such a condition. Well, we need monomers. And the monomer we're going to work with is nucleotides. They assemble into nucleic acids. Another monomer is amino acids. And we know from other people's work that these can form peptide bonds under these conditions and get all the way up into proteins, well, things we would call a protein. Amphiphilic compounds like fatty acids can self-assemble. They don't polymerize, but they self-assemble into membranous compartments. Those are the three main components that we have to find a way to make on the prebiotic Earth if we're going to get life started up. So I'm going to concentrate first on the lipids. There's what a fatty acid really looks like. That's one, I think it's probably about 16 carbons long. It's an acid because you can see a proton, a hydrogen ion coming off the end, and that's the definition. An acid is something that can release a hydrogen ion when it gets into water. Then you see today's lipids. That's a phosphatidylcholine on the left down below, and cholesterol. Those are the lipids that are in our membranes today. Two examples, in fact. And you can still see that they're amphiphilic. They got the hydrocarbon chains, the head groups, and that is what allows them to form membranes. Every one of you has made a membrane. You didn't know it at the time, but soap bubbles are amphiphilic molecules. Soap is an amphiphile. And when you blew soap bubbles, you were making membranes. Not exactly like what we make in a water solution, but membranes nonetheless. So here now you're going to actually see the formation of membranes from a dried lipid film. On the left is phosphatidylcholine, and on the right is water. Somebody doesn't like me up there. It's like the golden buzzer or whatever it is in Britain's Got Talent. Okay, so on the left is dry lipid, on the right is water, and watch what happens when water interacts. Now, some music goes with this. I use this in undergraduate teaching. I hope I've turned the music off, but maybe not. Let's see what happens. Aha. Now then, see those tubules coming out? That's very common. Those are multilamellar tubules, and they're unstable. And at the end of the tubules, you can see them breaking up into lipid vesicles. So that is what happens when you have dried amphiphilic molecules. And that's what we see under the microscope, the actual formation of lipid vesicles. It's really beautiful to watch. Here's what one of those membranes look like, if you can see it in cross section. Hydrocarbon chains in the center, hydrophilic groups on either side, and you can see a water molecules to the same size to give a sense of the size of these things. And you can see one little water molecule, it's that little red thing in the center there. Water does get across these membranes. It's small enough to fit between the chains. And when you take a drink of water, it goes across membranes freely. It doesn't need a transport mechanism. So now this is interesting. You look at that and you can say, well, that looks like lipid vesicles, but this was extracted from the Murchison meteorite. We did this way back in 1985, and we were able to follow the assembly of membranes from this extract of the Murchison meteorite. This stuff is 5 billion years or older than the Earth, in fact, and yet it still is able to self-assemble into membranous vesicles. We know that they're membranes because we were able to see this fluorescent dye that we trapped inside. And you can see these actually do have a permeability barrier so that they can hang on to a dye. So from that, I think that it's plausible that there were membranes on the early Earth self-assembling from stuff either being delivered or synthesized by a reaction called Fischer-Tropsch. It's another way that you can make fatty acids and fatty alcohols. So that is why I think that we can use, just as we say that, yes, amino acids were probably present, I think we can say that membrane forming compounds were present. And this would happen, maybe not in the ocean. Ocean salts would inhibit this kind of self-assembly, but at least in hydrothermal freshwater fields, you would be able to see this. Now here's something important that most people don't know. Suppose I have some molecules in solution, literally of almost any size from small di molecules up to ribonucleic acid. Those are the little round circles in this diagram to the left. Now suppose that I want to capture those easily into a lipid vesicle. What do I do? Well, it just turns out that a wet-dry cycle can also cause encapsulation. So if we dehydrate by adding lipid vesicles to a mixture of some solute, it could be virtually anything, and then put it through a drying process, the lipid vesicles fuse to form a multilamellar structure, and they capture those solute molecules between lipid layers. Then when you rehydrate, you get vesicles that contain the solute that you put in. This is the easiest way I know. In fact, when we got this to work back in the 1980s, you'll find my very first patent is how to capture big molecules like nucleic acids in lipid vesicles. Nobody ever licensed it, unfortunately, so that was too bad. Okay, here is something that any of you could do. All you need is some soap and some DNA. If you have some decanoic acid, which I know you guys have, and mix in some DNA, which I'll bet that somebody on campus has, put it through a wet-dry cycle, you have made protocells. It's that simple, a simple drying and rehydration. And now the DNA is inside the lipid vesicles and actually stabilizes them. It helps them maintain their stability. They've been stained with a fluorescent dye called acridine orange. It's a intercalating dye, so it gets in between the lipid layers, not the lipid layers, where the stacking of the DNA double helix. And that's what you're really seeing here. So it turns out to be very, very simple. Get all the way up to protocells. Okay, now we're getting to the last part of my talk. Can nucleic acids self-assemble? I've shown you that membranes can self-assemble from stuff like was in the Murchison meteorite. Well, I can tell you for a fact that adenine is in the Murchison too, about 1 part per million. Guanine is there, and that's from our friends at the Goddard Space Flight Center, Jason Dworkin and Glavin and Jamie Elsila have done this kind of work, working and showing that in fact the purines of nucleic acids are part of the mixture in the Murchison meteorite. So the question is, what are the source of nucleotides? Well, we can sort of take as a given that the bases were probably in solution on the prebiotic Earth because they were being delivered, if not synthesized by geochemistry. So somehow the bases became nucleotides. We don't know exactly how. Some very fine organic chemists, John Sutherland and Matthew Powner, are working on that. They do have ways to make it by organic chemistry, but I don't think anybody yet knows how it could be synthesized in prebiotic conditions. Then you can say what free energy drives polymerization reaction, which are basically condensation reactions. How can the entropy barrier, how can you overcome the disorder to get order out of disorder? How can nucleic acids resist hydrolytic hydrolysis? So, if we drive things uphill by removing water, if you add that water back, that's called hydrolysis. And those two oppose each other. You can drive things uphill by condensation. Hydrolysis brings it back downhill again, back toward equilibrium. How can we balance that so that you get an accumulation? Finally, how can nucleic acids evolve functions? That's where we are in our research. And I think that's where our field is. We don't yet know, even if we could make polynucleotides somehow, we don't know how to do that next really important step of selecting for function of the polymers in a protocell. So those are the questions. I am now going to tell you about steps 1 through 4. So the hypothesis, polymers resembling nucleic acids can be synthesized in freshwater hot springs. I'm going to quote Karl Popper. Karl Popper said, "A good hypothesis can be falsified." You heard that before, falsify? Now that's important because you should go into research, not with the idea of proving that you're right, that it's true. You should try to falsify something interesting that you see happening or an idea that you think is interesting. You should not hang on to the ideas. Most of them really don't map onto reality. Try to get rid of the bad ones and keep the good ones. And that's what Popper meant by falsification. So I'm going to show you a whole set of tests. And if one of them, I'll show you 7 tests. If one of them didn't work, I wouldn't be here now because the hypothesis would have been falsified. So I'm kind of going out on a limb, but I've been doing it now for 15 years. Here's what we're going to do. We're going to expose mononucleotides in conditions simulating the wet-dry cycles of hydrothermal fields. I have a carbon dioxide atmosphere, elevated temperature, acidic pH range, and hydration-dehydration cycles. We do that in an apparatus, and there it is. That's our simulation chamber. You don't need this big a deal, by the way. You can do this on microscope slides if you want to. It works. So here is an aluminum disc. It's got 24 wells in it, and each well can hold an experiment. The disc is rotated by a stepper motor underneath, and it's rotated so this well goes from here to here in about an hour. Then it goes back again. So it's about one revolution every 2 hours. So as it revolves, it goes through a flow of carbon dioxide coming from this tank, and over here it goes through it again. And right here, it has a hydration cycle. Water is dripping into the vial. Every time the disc stops rotating, water drifts into it there and there. And this is the volcanic heat simulation. We set these so that the disc goes up to about 80 degrees of temperature. So I got to show you one little chemical reaction before I go on. You've got to know how esterification works, how ester bonds form. So if you ever had organic chemistry, you may have done this experiment. If you mix ethanol with acetic acid, there's a spontaneous downhill reaction to form ethyl acetate. And it goes downhill until so much water has accumulated as a byproduct that it starts the back reaction, begins to balance the forward reaction. So that's called ester bond synthesis between an acidic group and an OH group on an alcohol in this case. What you have never thought of, I don't think, is that the same thing should happen with adenosine monophosphate. It has a acidic group, the phosphate group, right here it is. It's got an OH group. This is ribose. There's 2 H groups on the 2 position and 3 position. So if we can get this OH to come off the phosphorus and bind to that oxygen, kicking off that hydrogen, we have formed an ester bond. It should be spontaneous. That should go downhill. That's just really kinetics. That'd be a long reaction time, but you get a little bit of dinucleotide just by doing that. Well, the thing is, you've got to get rid of the water. If you want to pull this reaction to the right and make more and more ester bond, you've got to get rid of this water so that you don't have the back reaction. What we're doing in a wet-dry reaction, during the dry phase, we're getting rid of the water, and that pulls the reaction to the right. It's as simple as that. That's the chemical potential, the chemical energy that we're putting into this to get this stuff to polymerize. It also works with amino acid. This is the way you can make peptide bonds by heating and drying. It works, no doubt about it. So there we go. That's the core point. So what we're doing now is adding mononucleotides with or without a lipid matrix. Lipids are just there if you want to get protocells. You put the mononucleotides through multiple wet-dry cycles. That simply increase the amount of products. You get polymeric products as you can precipitate by the usual method of precipitating nucleic acid. And in this first experiment we did in 2008, we labeled with radioactive phosphate and did gel electrophoresis. So I'm going to show you that. It turns out that there are a couple of enzymes that are used to end label regular RNA. So if you've got RNA and you treat it with alkaline phosphatase to pop a phosphate off the end, then an enzyme called T4 kinase is able to take radioactive ATP and put a radioactive phosphate on the end of that molecule. So if we have made a polymer that resembles RNA, enzymes must work on it. This is a true falsification. If this hadn't worked, we'd be out of business. But it worked. Way back more than 10 years ago now, Sudha Rajamani in my lab was a postdoc, and we set this experiment up. And just take you through a few of these. Every one of these lanes is one separate experiment. Here we've done more and more cycles. One cycle, 3, 5, 6, and 7. And you see we get more and more yield. And look how long these things are. They go all the way from 10 mers up to over 100 mers in length, according to this ladder. These are known lengths of RNA that we put in to guide our estimate of length. Here's an actual known RNA species. It's very long, and so it tends to accumulate up near the top. We don't see all this stuff at the bottom. So here we've done it at room temperature at 60, 70, and 90 degrees. You get this reaction to go. Over here, we have different lipids present: phosphatidylcholine, phosphoric acid, lysophosphatidylcholine. And look there, we actually see a ladder. We are separating these polymers by single nucleotide resolution. So that was very important to us that we were able to see that separation. Then we did different ratios and different sorts of mixtures of molecules, AMP and UMP and so forth. So main point is it worked. So here's the next test. Well, it worked with radioactive labeling, but how do we know it's really like RNA? Well, if we can get a label like ethidium bromide, a fluorescent label, or this other one called CyberSafe, if we can add that and see fluorescence in the same range as that radioactive phosphate, that'll mean that this is like RNA, because that's what RNA does. RNA can absorb these intercalating dyes. So just to show you that point as a control, here is poly-U by itself, polyuridelic acid. It's a synthetic RNA. Here's poly-A, polyadenilic acid. And notice that there is nothing showing up if we just put in the polymer by itself, poly-U and poly-A. But if we mix them, we know for a fact that this produces a double helix. The poly-A winds up with the poly-U by Watson-Crick base pairing. And now that dye shows because there's an intercalating site. The dye can bind between the base pairs and the helix. And now the same concentration, we can see the dye fluorescing. And over here is our product. It is stained by this dye. So it looks like not only have we made a product, but it has a little bit of double helix in it, probably hairpins. We know for a fact that RNA just in solution winds up and makes little hairpins within a structure. Those then would intercalate these dyes. And here it is over here with another thing, CyberSafe. There it is, there's our product showing up very clearly. And here's a known double-stranded RNA polymer. So test number 2, it does absorb intercalating dyes. Now this is one that's going to take a little explanation. If we really are making a polymer, as I've shown you, we should be able to sequence it with nanopore sequencing. Nanopore sequencing, if you haven't heard of it, is something that basically was invented at my campus and my department. If you have a pore the size of a DNA strand, here's a little DNA strand, and if it goes into that pore, it's going to block the ionic current going through the pore. And if each base blocks a different amount of ionic current, we're going to get the sequence as that is translocated across. So it's as simple as that. So it is now a commercial instrument. You can buy one of these, get a MinION for $1,000. Is by far the least expensive way to get into sequencing by yourself. Typical machines by other competing manufacturers are $50,000 and up. So this is an easy way to get into sequencing. Furthermore, it's used every place you want because all you need is this. In fact, this laptop has the software to do it. I could plug a MinION into that and I'd be able to sequence DNA right here. It's been sequenced on the International Space Station. It followed the Ebola epidemic in Africa some years ago. Most recently, this is really interesting. The bigger version of this is called a Prometheon, and it got a human genome in 8 hours. New record for an entire 3 billion bases of the human genome. This thing also can sequence a length of DNA 1 million bases long. Nothing else can do that. This is absolutely a unique ability of nanopore sequencing. Well, the point is that I know enough about nanopore sequencing so I can do this test, but I want to show you how it works. You start out with a double helix of DNA, and a helicase is pulled into a nanopore along with a leader strand, and the helicase begins to unwind the DNA step by step at a rate of about 450 nucleotides per second. And as it goes through this pore, it interrupts the ionic current right there, this so-called limiting aperture. And this is a bacterial porin called CSGG, and it was developed by the company that does this. This porin is embedded in a membrane, and a voltage of 180 millivolts imposed to pull this in and down. And here is an actual trace of DNA being pulled through, and that's T, G, A, and so forth. So these are picoamp modulations of the ionic current. So we're going to see if we can actually get a sequence from the bases that we're making with our wet-dry cycle. That's kind of the ultimate test. But instead of RNA, we're going to use DNA because this nanopore is really specialized for DNA. So here we go. We're going to use deoxymononucleotides. We're going to use thymidine monophosphate. We're going to keep it simple, just one nucleotide. We're going to do multiple wet-dry cycles, and it's going to form, we hope, oligomers, thymidyllic acid. We're going to ligate that to a known DNA. The little bits pieces of oligomers we're going to be making are not long enough for nanopore sequencing. It takes about 500 nucleotides in length to really work. So we're attaching it by ligation to a much longer strand of known DNA. And then we put it through the nanopore. So this is what it looks like. That's our solution going into a MinION. And here is the signal of one DNA molecule going through this pore under the control of the helicase. This up and down is the bases modulating the current going through the pore. So that is what gives us the sequence. So this would be the DNA strand itself, this up and down. There's only about 4 different levels as you see here. Then suddenly it goes down to this, and that is the conductance that is specific for thymine, where the TMP is. And so you can see it all down here. So there is our oligo DT going through the pore. Then it comes unstuck, rattles around, and then exits the pore. So this all happens in just a few seconds. This goes through at 450 bases per second. When we look at the DNA that we made by gel electrophoresis, we can see each oligomer separated by radioactive phosphate labeling. So it starts down at 30 mers and goes up through 40, 50, 60, all the way up to 90 mers. And you can see that this is a scan of all of these little guys here showing up and telling you where this is. And look, over here, we can see the oligo T's now attached to the known DNA. Here's a short strand, a longer strand, and a very long strand. So it worked. We made DNA that is recognized by the enzyme responsible for nanopore sequencing, and we can see that DNA in the gel and in the base calling. So we're getting close to the end now, so hang in there. I just want to show you encapsulation. Here, what we've done is put a mixture of AMP and TMP. Those are base pairing mixture. We're staining the polymer with DAPI, which is a specialty stain for DNA. And the lipid is an artificial mixture of decanoic acid and decanoyl monoglyceride. And you can see the membranes that it makes just the lipid itself. So now we put this mixture through one wet-dry cycle, and we see a little bit of fluorescence accumulating. Here's 2 cycles, 3 cycles, and finally 4 cycles. And you can see this is the phase and this is the fluorescence. And down here we have the phase and the fluorescence. And you can see these accumulations of what we assume is the DNA in these vesicles. So it's just what I told you, that if we have an amphiphile present, we should capture, encapsulate. This is what we call protocells, okay? Okay, now let's see. Oh yeah, this is fun. I told you we're taking it out of the laboratory. We were in New Zealand 2 years ago, and what we did was to go to a hydrothermal field in New Zealand is near Rotorua. That's a little village in the North Island of New Zealand. And we perched ourselves. I think you can actually hear this boiling. Nope, I guess not. We sat on the edge of one of these pools, and you can see now all of the experiments we did. Each of these is one experiment, and they're numbered, of course, so we keep track of it. And they're in an aluminum holder. And we put this down into the heated pool. It's up around 91 degrees C. And then we use hydrothermal water from the pool to cycle. We did about 4 cycles, wet-dry cycles, half an hour each of all of those. We took it back to my lab in Santa Cruz, precipitated it, and measured the amount of RNA that we had made. So we did this experiment twice, once in 2018, sort of preliminary experiment, and once in 2020. That's the one I'm talking about. And we got similar results. If you take a look, you'll notice that UMP by itself was the one that gave us the most yield. You can see it here. Whereas when we had A or even A mix, we had less yield than all of this. And look, we got that same pattern here. Here's all the A's, AMPs, and various lipids. And now as we get down with more U's, we got more and more U's. So it's very comforting for us to know that we can make several hundred micrograms of a polymer product using real hot spring water. Furthermore, we could see it in gels. Here is what I showed you before. Here is the laboratory version of this experiment where we see the amphibian bromide labeling. And here's that same thing done with the hot spring conditions. And with several of these showed up very nicely on gels. So what we're doing in the laboratory also works out in the wilds. Okay, this is my last few series now. If we really are making polymers, we should be able to see them. Atomic force microscopy will let us see polymers of RNA. And you see those here. This is polyadenylate acid, that same poly A I showed you before by atomic force microscopy. This is synthetic, is made by an enzyme called polynucleotide phosphorylase. And look, you get some really long mers. There's about 1,000 mers. More likely, these are 50 mers to 100 mers, smaller pieces of this. But that's what RNA, known RNA, looks like. So we're going to show you now what we see. Well, one last control. We also wanted to have a true biological RNA, so we took viroids. Now, viroids are the smallest known infective unit. These things produce plant disease. They can be transmitted from cell to cell, and they kill off plants, some agricultural plants. And there's nothing more to them than about a 300 mers, much smaller than a virus. And you can see that they should be a rod-like structure. And here's what they look like. We did all of these with some viroids supplied to us by our French colleague, Marie-Christine Morel. And we can see these viroids. Here's some little viroid rings. Apparently, these hydrogen bonds can break, and we can see the ring structure of show up. Well, that's important because when we first looked at some of the preps that I made in my lab and sent to Tue Hassenkamp, he's my colleague at the University of Copenhagen in Denmark, he said, I'm seeing a lot of ring structures. I had no idea that we see anything like this. Here they are. If you look around, you see ring after ring after ring. So as soon as I saw that, I called my friend Ram Krishnamurti. He's a specialist in RNA chemistry. And he said, well, yes, RNA does form rings. Some of them are biological rings. So if you make RNA synthetically, very often the ends show up with each other as a ligation point, and they make a ring. So we're very pleased by that. We also see that some of them have these thinner strands attached. We don't quite know what to make of this. It could be that it makes a long strand and then somehow one end cyclizes. You can see that happening here. So that's some of the stuff that we make. We can actually see these as rings. Here's some more examples of rings. This compares GC, which is GMP plus CMP, that's a base pairing. Here's AU, AMP plus UMP. And they both make these ring structures. Single nucleotides did not form rings. There's something about having base pairing present that helps these rings to form. We don't know why. It's just an observation at this point, but I think it might be quite important that you do have to have base pairing possibly. Okay, there's one last thing I want to show you. GMP is a special case. It's the only nucleotide that forms what is called a quadruplex. There's GMP, there's the guanine, there's the ribose, here's the phosphate, and this little abbreviation. You can see this tetrad structure referred to as a quadruplex. These tetrads can stack into a fiber that you can see by atomic force microscopy. So this is not covalently linked. This is just the stacking energy. There they are. Here's an atomic force microscopy of GMP plus CMP dried down at room temperature. The GMP has formed those tetrads, and they have lined up on the crystal structure of the mica underlying this film. So that's what it looks like if you just dry down GMP plus CMP at room temperature. But this is what they look like. By the way, we've sent this off for publication. You're my first audience to see this. Here's what they look like if we cycle it at 80 degrees C. We see aggregates, and off of those aggregates, we see what appear to be polymers, thousands of nucleotides in length. Somehow, having the base pairs present and having The cycling and the heat allows G plus C to form covalent bonds and produce these really long RNA strands. We're going to get a lot of referee critical comments on this. It's going to be pretty controversial because we don't have anything else other than AFM to say those are real, but they look real to us. We've done all the controls. We only see those in just what I've told you here. There's got to be cycling, there's got to be heat, and there's got to be wet-dry cycles on mica. So that's the end of my story. And I'm going to tell you what I told you. Here's an alternative scenario from hydrothermal vents. Life began in freshwater hydrothermal pools subjected to wet-dry cycles. Life did not invent nucleic acids. Nucleic acids can self-assemble all by themselves, just as I've shown you. If amphiphiles are present, you get these protocells. The protocells are all different from each other. They're experiments. The variations allow them to undergo selection, and I hope that in the near future, we will see them begin to evolve. Thank you.
Okay, so I'm sure Dave is open for questions, so he has some questions. Yes, please.
Yeah. So I have a couple, three. So feel free to answer whichever one you need to.
Just one thing.
Sure.
We're gonna have trouble hearing you.
Yeah, can you speak up?
The first question, the part which we were talking about, research methods in general, and then the idea how hypotheses should be falsified,
Yeah.
right? But then, however, two subsequent kind of ideas came into my mind that inherent in the current state of the interdisciplinary field of astrobiology, is it possible that the fundamental nature of the research data cycle could be fulfilled in this way, and that we could ultimately falsify hypotheses when there's a lot of conjecture and a lot of very rigorously controlled and the best conjecture we have in the data and technology at this time, that we could fulfill this. And then the sum of that would be what is the extent that we can operationalize conceptual variables to the best of our ability that gets at the research methods. So I just to verify what we have.
Sure, I got the gist of the question. So, weight of evidence.
Yes.
We have to be critical about our results.
Yes.
And not depend just on one thing.
Yes.
So I've told you, these linear structures we see, we don't have that second independent test, that those are really polymers. All we do is we see them. So that's interesting. They might be polymers. I think they probably are. We're going to have to get a second test.
Yes.
Because that's a falsifiable thing.
Is it a second test from your own lab group or from ones that are attempting to do the same but are independently run?
Okay.
You know what I mean?
Let me tell you, there is one secondary test that we have run.
Okay.
Maybe you and I can come up with a prediction.
Of course.
Here is the prediction. We know for a fact that the UV absorbance of the double helix increases if you melt it. So here's the double helix, my little fingers, right?
Yes.
You heat it up, vibrates, it comes apart. And it's extraordinary. It's called hyperchronicity. The UV absorbance increases when it comes apart. Hyperchronicity. So that's a prediction. If I have made, using base pairs, covalent bonded stuff,
Yes.
it should come apart, and I should be able to see hyperchronicity in that preparation. So we've done that once, but only with AMP and UMP, and never with anything that we've seen by atomic force microscopy. So, so even though that sort of work, we did see hydrochronicity, we're still not certain. So that's the way science really works.
Yes.
I think all of science works, right? So we are all jurors, we're all judges, and we're also all lawyers arguing our case. That's a pretty good metaphor for how we tend to think in terms of explanatory power. A good hypothesis with testing has explanatory power beyond its alternate hypothesis.
Thank you. Yeah, I have two more, but let's go ahead and let others go ahead, and then if there's time. Okay.
I have a question. Hello.
There's a question
Get down the stairs.
on Zoom.
I've been around science a long time.
Hello, I have a question.
Go ahead on Zoom.
This is Yok Yong from Caltech.
Hi,
Hi.
do I know your name?
Yeah. Thank you for giving a fantastic seminar. I enjoy it. I have two questions. The one is that 60 years ago, Fox did similar experiment with amino acids, not with this nucleotide. So, so are you sort of doing your experiments at the parallel of Fox's experiment, but using a nucleotide instead of amino acids?
Actually, I knew Sid Fox, and we often would joust in meetings about this. Sid heated his amino acids to temperatures at which bonds other than peptide bonds could form. So his proteinoids were actually had some peptide bonds, but they also had other bonds just because he heated to, I think, about 160 degrees. We're staying below the boiling point of water. So we can get ester bond formation preferentially without activating other bond formation. So Sid began to try to prove that he was right. He really believed that he was on the right path. And when it turned out that a lot of his bonds were something other than true peptide bonds, people's interest kind of waned in that approach. But you are right saying that both Sid and I are using heat energy as an activation energy source. We both are reducing water activity to inject chemical energy into it. But he injected so much activation energy that he began to activate other bonds than peptide bonds.
Okay, yes. So the difference is between his 160 C and your 80 C. So I'm talking about a factor of 2 in temperature. So that is the big difference. Yes, I understand that. Thank you. But then I also, Cece, I have another question related. If your temperature is so low and your bonds are fairly weak, so when you put the water back, why don't you everything hydrolyze and just go back to where it came?
It's a lovely question because I can answer it. Okay, it's called a kinetic trap. A kinetic trap in chemistry and organic chemistry in particular is when an uphill reaction can go quickly and the downhill reaction goes slowly. Hydrolysis at these temperatures is much slower than the uphill synthesis. So that means that the polymers accumulate in a steady state away from equilibrium, and you don't have an immediate downhill reverse of the reaction.
Oh, I see. So it's not a steady state. It's not an equilibrium state the way that you have.
That's right.
Because
That's right.
the kinetic rate coefficients are different.
Yes, I began, as I was thinking about that, I began to realize that all life exists in a kinetic trap when you think about it. We synthesize our polymers quickly and they hydrolyze slowly. We don't hydrolyze when we take a shower, at least very much.
Fantastic. Thank you very much for answering
Thanks for the question.
my questions.
We're running a bit over time here, so are there any other questions? Maybe one? Zach.
Thanks, Dave. There's so much really interesting stuff here. I have a couple questions, but I'll just ask one right now. The AFM experiments that you showed us at the very end, were those done dry or in solution?
Oh, yeah. So the way it works, question is, how do we do the AFM? They were done on a freshly cleaved mica surface. This is the way you do AFM, that's the surface. Then we would put the mixture on it, just maybe 20 microliters of the solution. It would dry, and we add water back, that's the wet cycle. They would dry again, wet, dry, and then you would flush it with water to get rid of all the excess monomers, as well as smaller molecules that would dissolve off. But as these polymers got longer and longer and longer, they had more and more adhesion to the mica surface. So that's why we don't see them in ordinary experiments where we simply just do this in a test tube. I think we're losing all that long stuff because it's so sticky. You know, we simply don't get it off out of the test tube. Does that answer your question?
Yeah, that does answer the question. A quick follow-up about that. Is it possible to have, you talk about the mixture of non-covalent polymers?
Oh yeah, that's what the quadruplexes do. Those are non-covalent. So that is an example of a stabilized linear structure that is not covalently bonded. And it's long enough and stable enough so it really does stick to the mica as well. But maybe you're asking, how do I know that those long things coming out of the GMP and CMP mix are really polymers? Could they possibly be just, you know, little base pairing, for instance, as opposed to covalent bonding? We do have a test for that. We haven't done it yet. If they really are RNA, they should be attacked by enzymes. So we can put in an RNase and we should see them either disappear or just kind of turn into pieces.
Got it. Thank you.
Mm-hmm.
Okay, well, thanks very much for a very interesting talk, and let's all thank Dave.